go ahead with the next speaker. We'll just have him do his introductory slides really slow. So our next speaker is my co-organizer, Dana Spence, and he'll be talking about improved understanding of nitric oxide bioavailability using novel tools to control and detect oxygen levels.
Okay, we're going to try to find that. Okay, thanks, Sue, for helping organize the session and for doing most of the work presiding. Thanks, everybody, for being here. I have a little bit of a cough, so hopefully I can make it through without coughing into the microphone. So in keeping with today's theme of trying to understand or better understand reactive nitrogen and oxygen species, the good part, the bad parts, I think you'll find in my talk today that my good aspect or beneficial aspect is looking at the nitric oxide levels that we see primarily in the bloodstream. Again, I think most physiologists would view this small gaseous signaling molecule as a beneficial molecule in vivo. However, as we've seen from Mark's talk today, it can be used as a biomarker for a very, very serious condition. Our group is also interested in elevated nitric oxide levels in multiple sclerosis, which has been found in cerebral spinal fluid and other fluids in patients with MS to have higher levels of really NO metabolites, not necessarily NO, but that's something that we're interested in as well. However, the bad part, I think, about my talk today, which hopefully there's not too many, will be actually trying to explain why certain patients have actual low levels of nitric oxide. So that's one of the sort of underlying themes of my group for the past few years, is trying to determine why are these nitric oxide bioavailability levels low. And so one of the things I'm going to slide in the middle of my talk today is some work that Steve Halpin and Jada Urkel has been working on in my group, trying to develop new tools to better control and determine oxygen levels that may actually be perturbing nitric oxide levels in our system. So I'll talk a little bit about some very preliminary data that we have on that. And so what I'll try to do today specifically is to follow this overall outline here as a guide to my talk to try to keep me on track. I'm going to first introduce this new concept of ANOVA, which I read about in a journal recently, trying to explain some of the nitric oxide availability levels in vivo. I'll also talk about previous efforts by our group to actually measure nitric oxide using various techniques such as amperometry and fluorescence. And then, as I mentioned, I'll talk about some current technologies in our lab that are under development to try to, again, help us better understand what's happening in vivo, but performing these measurements on a controlled in vitro platform. And then finally, if I have some time, I'll talk about some upcoming studies involving nitric oxide with some work in our group involving sickle cell, and then also shown here some stored blood applications that my group has recently been investigating. And so first of all, I'd like to introduce this concept of ANOVA, or insufficient nitric oxide bioavailability. Again, I first read about it about 10 months ago in an article by Roback in the journal Transfusion. There was a special issue in Transfusion on red blood cells and some of the problems with storing red blood cells. And again, I think Mark actually alluded to in his talk, there's a lot of different theories floating around as to where nitric oxide originates in vivo. And again, there are some groups who believe that the red blood cell itself is somewhat of a nitric oxide donor in various forms. And again, those particular groups, the Stamler group and the Gladwin group, both had articles in this particular issue of Transfusion. However, I was sort of interested in this article here by Roback because what they're actually proposing in their article is that in addition to the red blood cell donations, if you will, of nitric oxide in vivo, that you sort of see like this overall lacking of nitric oxide, not only from the red cell, but in other tissues in vivo. And so this hypothesis was actually developed to help explain why patients who receive a transfusion actually have increased risk of morbidity as opposed to those people who don't receive transfusions under times of maybe surgery or some sort of trauma. And again, this particular theory, as the authors described it, was trying to be a little bit more universal or central in that they're trying to describe the overall reduced nitric oxide levels in various tissues. And so I found it very interesting because it's something that my group's actually been working on for a few years, is actually trying to describe what are the overall sources of nitric oxide, especially in the bloodstream. Are you getting like an echo on the microphone, or is that just me up here? I'm getting a lot of static. I'm going to put this down here a little bit. Okay. And again, as I mentioned, reduced nitric oxide levels have been reported in various disease states. Again, Randy Sprague's group at St. Louis University has shown that people that have primary pulmonary hypertension, or basically hypertension in the lung bed, have reduced amounts of NO in the lung. Coinciding with that, patients that have cystic fibrosis, they've also been shown to have low nitric oxide bioavailability. Diabetes, patients with diabetes are known to have nitric oxide levels that are somewhat lower, not only in the bloodstream, but in the endothelial tissues as well. And then, of course, patients that have sickle cell also have problems with overall nitric oxide in their bloodstream. And so I'm going to actually try to tackle a couple of these during the talk today. So why do we have this reduced levels of nitric oxide? Well, again, that's a question that I believe personally has many answers. Okay, and again, I know that for those of you who have seen me speak before, you're probably sick of seeing this little movie. But again, I knew there'd be students here that maybe haven't seen this particular little clip. So I got this film clip about 10 years ago from Mary Ellsworth, who's in the Department of Pharmacological and Physiological Sciences at St. Louis University. And what this is right here, I'm going to start this movie in a second, is this is actually tissue from a hamster cheek retractor muscle. And you can't see it too well right now, but when I start the movie, you'll be able to see it. There's actually this little line right here, and there's a line right there. That's actually a blood vessel in this hamster cheek muscle. And when I start the film, you're going to see these little black dots flowing through there. Those are actually red blood cells. And what Mary's group did in real time is they actually made the tissue surrounding that vessel hypoxic, meaning it had a need for oxygen. Okay? And what you'll see then is this incredible flux of red blood cells following a dilation of the blood vessel. And of course, the main job of the red blood cell in vivo is to deliver oxygen to tissues that need that oxygen. And so you'll see this red cell flux followed by a constriction after that demand has been met. So I'll start this up here, and then, so here you can see, this over here by the way is a vein, but here you can see the vessel. You make the tissue around here hypoxic, and you'll actually then begin to see the vessel dilate. So this is a real-time dilation, and then you'll also see this incredible flux of red blood cells, again, bringing oxygen to that surrounding tissue. Once that demand has been met, the vessel actually has the ability to contract, slow down the overall blood flow to that particular tissue. And in a moment, you'll actually see the tissue being, well, you won't see it being made hypoxic, but Mary made this hypoxic again. This isn't a repeat. It was actually all in real time. And so you'll actually see the dilation again. And this time I actually think the flux is somewhat higher than in the first one. And so this movie right here has actually been a key driving force of some of my group's efforts for nearly a decade now, because, as I mentioned before, there are many answers, I think, as to how is this nitric oxide produced? By the way, I should mention that one aspect of that dilation that you saw here, this blood vessel, is that nitric oxide is a very important determinant, a major determinant in that overall vessel dilation. It relaxes the smooth muscle around the vessel, allowing for the vessel to open. Why the reduced nitric oxide? Well, in people who have blood flow problems, they generally do have reduced nitric oxide. And one of the easiest answers, and you hear all the time, is people just saying, well, they obviously have some sort of endothelial dysfunction. Their endothelium, one of the main producers of the nitric oxide, isn't working properly, and that's why you're seeing the reduced NO. And in many cases, that's probably correct. Another possibility is that we have certain cells flowing through our bloodstream, let's say, for example, red cells, that don't respond to the local environment in an appropriate manner. And what I mean by that, as I alluded to earlier, it's pretty well established by many groups that the red blood cell, when exposed to a hypoxic environment, will actually release, I've actually measured micromolar amounts of nitric oxide into the bloodstream. In addition, and it's also been shown in certain cases that certain red cells don't release the appropriate amounts of nitric oxide when exposed to hypoxia. Now, another reason, and this is one that my group actually studies quite a bit, is that in addition to releasing nitric oxide, the red blood cells also release ATP, which is a very important molecule in purinergic receptor signaling. And when the ATP binds to purinergic receptors on the endothelial cells will then begin to produce nitric oxide through an eNOS-based mechanism. And so it could also be a problem that certain cell types don't release enough, let's say, ATP in response to certain hypoxia, which has been shown to be the case, at least in patients with cystic fibrosis, the pulmonary hypertension, and the diabetes. Okay. So again, this is one of the things that my group then has been trying to answer for a number of years, is trying to tease out what is the main culprit of the reduced NO. Is it the lowered ATP? Is it the, you know, the reduced release of nitric oxide itself from the red cells? And again, as Mark alluded to earlier, there are other mechanisms by which our body can produce NO. These are not the only 2 or 3 that I'm showing here, but these are the ones that we investigate. Okay, so our group has, like I said, long had an interest in measuring nitric oxide and ATP release from red cells and endothelial cells, et cetera. And so this right here, I actually dug this out last night. This was one of our first attempts to measure nitric oxide from endothelial cells. And so this was due in large part to efforts from Scott Martin's group when we were both at St. Louis University. What we did in this particular case, and I'm abbreviating a lot of the work here, but with Scott, well, and this is largely, like I said, Scott's group, he would take a glass slide and using a PDMS slab that had a channel in it, he would basically pull carbon ink through that channel, bake it / dry it, and then pull that PDMS slab off the glass, and what he'd be left here with is sort of this small micron size carbon electrode, this carbon ink electrode, if you will. Our group would then take another piece of PDMS, which we had layered a confluent layer of endothelial cells in this particular channel, maybe 100 micrometers wide by 100 micrometers deep, and we'd lay that channel over that carbon ink electrode, stimulate these cells somewhere downstream, maybe with ATP, maybe with some sort of calcium ionophore, to get a nitric oxide release from these endothelial cells, and when pushed due to maybe some sort of syringe pump over these electrodes, we could actually measure nitric oxide spikes coming from the endothelial cells. And so this was actually published in the Analyst, I think, back in 2004. So this here is about 8 years old, this data that I'm showing you here, or at least this image. And again, we were really excited about this at the time because it enabled a direct measurement of nitric oxide. We didn't have to modify really the cells under investigation. We could simply use, you know, some sort of stimulus. And also we could perform repeated stimuli on the same cell type. So these are some of the things that I really liked about our amperometric techniques. However, after I moved north, I found that, and I think basically it's because I wasn't around Scott's labs anymore, is that our success rate went down using the amperometric techniques. So I think what this is saying is I'm just not a very good electrochemist. But I also wanted to move towards higher throughput measurements. And also I found that it was difficult to get at the intracellular values of the nitric oxide without perturbing the cell itself. And I didn't really want to do that. And so when I got up to Wayne State, one of my postdocs, Paul Root, told me that when he was at Windsor earning his PhD, he had measured nitric oxide using the fluorescent probe, the DAF-FM family of fluorescent probes, okay, which again we heard about in one of our earlier talks. And so this is how we'd been measuring the nitric oxide in our microfluidic devices using these DAF-FM probes. And again, real quickly here, I'll show you what we do is we start off with a PDMS slab down here, and these L-shaped objects here, or features, are actually our channels. And then what we do over those channels is we actually put a thin, porous polycarbonate membrane over those channels. We then bring in another piece of PDMS, which is essentially featureless except for the holes that are punched out here with a paper hole punch. And we essentially make a sandwich of the PDMS with the polycarbonate in between. What we can do then is on top of this membrane, we can either leave it blank, we can load these wells with the DAF-FM probe, or we can actually culture endothelial cells on the top of that membrane. Meanwhile, flowing are red blood cells that we might subject to some sort of hypoxic environment, a pharmaceutical drug of interest, etc. And so what we have here in this particular situation is a way to sort of mimic the bloodstream and the cell-to-cell interactions that might be occurring between red blood cells and the endothelium. Okay? And so we've used this technique to measure nitric oxide from the endothelial cells and the release directly from the red cells, as I'll show you in the next few slides. For me, this is easier than the electrochemical methods. I like using the fluorescent probes, although, you know, as I'll mention in a minute, they have some drawbacks. The inter and intracellular values of nitric oxide are easy to obtain, and also it enables microscopic imaging. And as one of the things that our group just published a couple years ago, we can now actually take these devices, and if we make these wells the same distance apart as your classic 96 well plate, we can pump our blood through here, get the nitric oxide signaling going, and we put it in the plate reader and walk away. So we actually have plate reader determinations here of our microfluidic devices. So again, it truly does start to approach high throughput. Again, Not without its problems. I don't like putting the probes into the cells because, again, now I'm perturbing the actual cellular system. So some cell manipulation is required, which, again, is not the case with most electrochemical techniques. The other thing that I don't like is, I mentioned with the amperometric system, I can perturb my cells time and time again and measure nitric oxide release. And when you use the fluorescent probes, it's sort of a one-shot deal. You add your probe, you stimulate, you measure the fluorescence, and you have to move on to a different well. You're done. So again, some disadvantages with the fluorescent system. But we have used this to learn a few things out, determine a few facts about the sources of that nitric oxide. And so a couple years ago, I think it was, we actually set out to start differentiating where's this nitric oxide coming from. Is it directly released from the red blood cells? Is it ATP being released from the red cells that are stimulating the endothelium NO? And again, keep in mind, this isn't even talking about the macrophages and platelets and all the other nitric oxide generators in the bloodstream. So we're definitely reducing this system down to try to make it something that we can understand. So one of the first things that we did is we actually wanted to look at the red blood cell release of nitric oxide. It's something that our group had never really done. And we tried it in microtubes. And so what I mean by this is we actually had a deoxygenated buffer. We'd add our red cells, spin it down, take off the supernate, and then see how much nitric oxide was in there. And again, we've heard time and time again in this session that nitric oxide isn't your most stable molecule, and so you knew that this was going to have some problems involved with it because, again, how much of our nitric oxide was actually just being oxidized and changed to another form. When we did that, we could actually see about 2.1 micromolar nitric oxide released from the red blood cells, but again, we weren't too confident in that value. And so what we did then is we went to our microfluidic systems where, again, I'm showing you a side view of the system that we've employed. We actually will push our red blood cells through the channel. This beige thing here is actually our polycarbonate membrane. And on top of that membrane, we have our DAF-FM probe, which then what we do is the red blood cells flow through here. If they're made hypoxic, typically offline at this point, they will release the nitric oxide, which will diffuse up through the porous membrane, hit the fluorogenic probe, and we'll see a fluorescence, which we can measure. Okay? And when we do that, one of the things you'll note here is that our normoxic RBCs flowing through the system, I knew I'd get your head at one point, we're releasing, you're okay, I'll go over you. We're releasing, we'd find less than 1 micromolar nitric oxide being released from those RBCs. However, in using our microfluidic device, we are now detecting over 7 micromolar amounts of nitric oxide. So 3 to 4 times the amount than when we did that microtubes assay. And so again, I was actually somewhat surprised by this. I mean, I'd read all the literature that the red blood cells release copious amounts of NO, but again, sort of measuring here is believing. These red blood cells do respond to hypoxia and release quite a bit of nitric oxide. We did want to move forward, though, because again, even though you see the red blood cells releasing this amount of nitric oxide, as I mentioned about the stability of this molecule, I still had a problem believing that released NO, be it nitric oxide or in the form of a nitrosyl-thiol, etc., could make it through the plasma, through the platelet layer, through the endothelial layer to the smooth muscle cells where it would participate in vasodilation. And so I wanted to keep using our devices to learn more about the nitric oxide that was actually above that endothelial layer in our device, meaning that NO that would participate in the vessel relaxation. And so one of the things we did is we also used our device here to look at how much ATP was released from these red blood cells when subjected to hypoxia. Because again, remember, this is going to set up a future slide here where we're looking at how much nitric oxide was stimulated by the ATP. Okay? And so you can see here normalized ATP release using a chemiluminescent luciferin-luciferase assay for ATP. Here's our red blood cells. We make them hypoxic. Again, you see about a 50 to 60% increase in the amount of ATP that's released. And we can actually shut that ATP release off by either using diamide, as shown here, which is an ATP release inhibitor because it makes the membranes more stiffened. We can also use things like niflumic acid or glibenclamide, a CFTR protein inhibitor, to turn off the ATP release. So again, this slide here just showing that we can measure the amount of ATP that's released from these red cells when exposed to hypoxia. Now, again, getting to our question though, what's the NO that we're seeing above the endothelium? Is it RBC derived or is it stimulated by ATP that is released from the red blood cells? In that case, endothelium derived. And so one of the things that Steve did in our group is we wanted to see how much nitric oxide could actually get through an endothelial layer. And so what we've done in this particular case, if you focus on the little picture down here, again, we have our polycarbonate membrane, we have our channel flowing underneath, But this little pink layer here actually represents a layer of endothelial cells that we immobilize on top of the membrane. And again, then we have our fluorogenic probe on top of that. But what we've done then is we've actually flowed various concentrations or pumped various concentrations of a nitric oxide donor molecule through the channels to see how much would actually get through the membrane and in some cases through the endothelial layer. And so the dark bars here are actually showing the results from when we pump a 9 micromolar nitric oxide donor through this device here, but without any endothelial cells on top of the membrane. And for the most part, you can see if we pump 9, we measure about 9. If we pump 7, we see about 7, etc. When we have endothelial cells on that membrane and pump the same solutions through there, above those cells, if we pump 9, we don't even hardly see a micromolar. Same thing for 7 and 5. What this is suggesting then here is that when you have the endothelial layer on that membrane and you pump a nitric oxide donor underneath it, if you have your probe on top of those endothelial cells, not in the endothelium, you don't measure hardly any nitric oxide above that endothelial layer, suggesting that the NO is not getting through the endothelium. Okay? And again, the reason that that's important then is that if we move to a system like this, where now what we're doing is pumping our red blood cells, okay, under an endothelial layer, shown here, but now instead of pumping NO standards, we're actually going to pump red blood cells. What we see then here is that if we're measuring the nitric oxide above the cells, okay, and let's focus on the black bars here first. So this is the nitric oxide production above our endothelium here, okay, above the endothelium. We move red blood cells through there. We see a little bit of nitric oxide above the endothelium. If we make those cells hypoxic, look how much nitric oxide now we see above that endothelium. Now again, the question becomes, where did that NO come from? Was it from the red blood cells releasing NO and made it all the way through, which our standards show probably can't happen? Or is it because the red cells released ATP that stimulated the NO? If we shut off the ATP with that diamide, remember I showed you that in a couple slides prior, we shut off the ATP release, again, now we see a value of nitric oxide that significantly decreased from when the red cells were just allowed to be hypoxic without the diamide. Okay, and again, without any hypoxia, we don't see any nitric oxide at all. So again, what this data suggests here, and by the way, we also ran the same studies by using a nitric oxide production inhibitor in the endothelium to show then that with this bar right here, when we do make those endothelial cells incapable or at least reduce their nitric oxide production, you don't see the nitric oxide above the cells. So again, what this suggests here largely, or I think at least strongly, is that the nitric oxide that you're seeing participating in vessel dilation is actually endothelium derived and stimulated by ATP release from the hypoxic RBCs. Okay? So these are some of the kind of measurements then that we can make on these controlled in vitro platforms. However, we do have one problem here with our system, and this is, again, why you get new grad students and you challenge them to do new things. We made our systems hypoxic using this oxyrase, which is, again, I don't even really know what it is. It's an enzyme that basically chews up all the oxygen in your system. We don't know what it's doing. We don't really have good control of it. And so what that means then is that in a lot of the studies I just showed you, we did make the system hypoxic. However, it's a very binary system. Our systems are either sort of saturated, room oxygen, or they're almost anoxic. And in vivo, that's not going to happen that often. In fact, if you see here, Jada made this nice slide here showing the various oxygen tensions of, like the air in your trachea, alveoli, and then also blood returning to the heart. There's a wide range of oxygen saturation levels in the bloodstream. And I want to be able to study the cell function, the red cell function, the endothelial function, et cetera, at these various oxygen tensions. And so this is what some of our students in the group have been working on, is making a microfluidic device now that enables better cellular control. Okay, and so what we actually want to have here then is, again, a device similar to what I just showed you. Red blood cell samples coming in here, flowing through. I got our membrane right here. Maybe one well with luciferin luciferase to measure ATP. Another one here to measure the fluorescence from the DAF-FM and the nitric oxide. Again, we can do this all in the plate reader now, so it enables these multimodal detection systems to be relatively easy to perform. However, here I actually want to have an oxygen control region. Okay. What this means is I want to be able to flow red blood cells over maybe a solution of sulfite, which will deoxygenate that solution flowing over that region. However, just centimeters downstream, I want to have some electrode system here that can tell me what is the oxygen tension right after we deoxygenate the system to a certain extent. And then after we know this oxygen tension, the red cells will flow up and we can measure the resultant release of the molecules from the cells. And so this is how we've been approaching this so far. And again, as I mentioned at the beginning of my talk, this is very preliminary work. Again, stealing data from Scott's group at St. Louis U, we've actually made substrates out of the C7 epoxy. And what we do here, again, borrowing technology from Scott's group, we can actually put in gold working electrode wires and silver chloride electrodes here to serve as an electrode system in this epoxy. And again, if you haven't read these papers from Scott, you should take a look at them because they're fantastic. You can put these metal wires in these these epoxy matrices here, these bases, use them, and at the end of the day, you just polish them and you use them again the next day. The other great thing is that our group can actually take thin layers of PDMS here and actually lay over these electrodes, serving as a membrane to protect the electrode system from, as Mark mentioned, the blood. You got to do something to try to help out your electrode systems when you're actually flowing blood over these electrodes. And so that's what we've done here is we've used these epoxy systems with the electrodes. This channel right here is actually also in the epoxy. It's eventually going to represent our oxygen depletion region. And then in this thin PDMS layer, we actually have what amounts to a little cavity here, has some volume in it such that when we put an electrolyte solution over our electrodes and then seal this membrane right here over it, we actually have some volume so that the electrolyte solution can sit right here in this little notch made in our PDMS membrane. We then place another layer of PDMS on that with a waste well and an inlet, and what we've actually been able to do now is, to this point, pump red blood cells over these electrode systems that have either been made hypoxic or normoxic, and we can actually detect the oxygen levels over these electrodes. And again, here's just showing, using soft lithography, how we make the PDMS. And again, just, a little bit of a technique here on how we make our epoxy rings. It's actually quite easy. You use a glass slide with some sort of retaining layer. You fill this with the C7 epoxy and the Armstrong activator, bake it overnight, and you can actually pull that epoxy mold out of there. And our group actually drills the epoxy afterwards and puts the metal wires in there and then backfills. My students told me that you can keep bubbles out of it a lot easier if you do it that way. So again, very simplistic to make. We had an undergrad, I think, doing some of this work as well last year with a lot of success. So again, a nice simple technique. Here's some of our initial data. Again, this was just some of the buffer here, oxygenated buffer flowing through the system. The spikes here, I think, is when Jada may have actually touched the outlet with like a chem wipe. Is that what those are? Yes? Yeah, dabbing. Okay. So these are just noise spikes here. But then when she actually injects hypoxic red blood cells, which were, again, made hypoxic offline in this case. We're not to the point where we're actually deoxygenating online, but we're getting there. We can actually see decreases in our overall oxygen signals, as one might expect from making this simple, rather hypoxic. Okay, so we're getting there. What do I have, about 7-8 minutes left? Is that right? Okay. So with some of these technologies, I just want to talk here real quickly at the end about how we're using them, and getting back to this whole concept of ANOVA, the insufficient nitric oxide bioavailability. And again, I mentioned sickle cell, so maybe I'll take just a moment here and explain why we're interested in sickle cell. Again, autosomal recessive disorder. You can see the numbers here of how many people typically have sickle cell or sickle cell trait. Again, formed from a mutated hemoglobin, the HbS, which again, if you become deoxygenated, that hemoglobin will begin to polymerize after a few minutes. If it actually reaches full polymerization, after those 3 or 4 minutes, the cell will probably start to sickle. If you can get the cell reoxygenated within a minute or two, while the sort of the nucleation, before full polymerization, the cell will actually reverse and not completely sickle. So it's important, if you do have a deoxygenated red cell that's prone to sickle cell trait, if it becomes deoxygenated, if you get it reoxygenated back into the lungs, you're going to be okay. Because what happens here, if you take a look at the circulation as you leave the cardiac here, you'll actually flow through oxygenated. It goes through the various tissues and organs, the kidney, skeletal muscle, the liver, etc. But as it goes through these organs, it becomes deoxygenated. And so this is sort of your danger point here. You've got to make it back up to the right side of the heart and to the lungs where you get reoxygenated before that polymerization sets in. Okay? So again, you can imagine then that nitric oxide production in the sickle cell patients would have to be key, because again, if you can get the good blood flow going after the deoxygenation here, you can make it back up to the lung bed and get reoxygenated before the polymerization sets in. And so one of the drugs that's often used to help reduce the sickling crises, in fact, it's the only proven therapy for sickle cell, is Hydroxyurea. Again, it lowers the rate of sickling crises. It increases the polymerization time frame, so again, it gives you more time to get up to that lung bed. The interesting thing about hydroxyurea, though, is that its mechanism of action is kind of unknown. Most people will tell you that it increases fetal hemoglobin levels, which then holds on to the oxygen more tightly, and you don't have the deoxygenated heme. However, there's a lot of people that believe that there has to be some other mechanism, because when you take hydroxyurea, you actually see beneficial effects in your blood flow hours after taking it. It's just an oral, tasteless medication. Okay? However, HbF increases aren't observed in these patients, sometimes for 3, 4, 5 months, if at all. The increase in HbF is only seen in about 30 to 40% of patients on hydroxyurea. But it's a very successful therapy. And so recently, I don't think it's on here, and I didn't put it in this particular talk, our group had actually shown and published it in the European Journal of Pharmacology back in the summer of 2010, that if you take red blood cells, at this time we were working with rabbit red cells, but we've shown this on humans, if you add hydroxyurea to these red cells, it actually stimulates a little bit of nitric oxide in the red cell itself. The red cell, even though it doesn't have any nucleus or mitochondria, it does have a little bit of enos in it. So it'll produce a little bit of nitric oxide, and again, we don't know the complete mechanism yet, but after that nitric oxide has been formed, after 30, 40 minutes, if you flow those red blood cells as it would in vivo, it creates enough shear on these red cells that the ATP release from the cell now goes up 50%. And we showed that in that paper in EJP. But the one thing that I'd never done, though, is shown the downstream effect of that ATP. And so when Sarah Lockwood joined my group, at the same time Jada did, I put them on this project here, and I said, let's see if we can finish this little in vitro study up and show that that ATP is actually resulting in a nitric oxide increase in the endothelium. And so just recently, Sarah has used sort of our workhorse microfluidic device here to actually show the interactions between the red cell-derived ATP and the endothelium NO shown here. And she's found some really interesting results. If we look at the normalized fluorescence intensity from our endothelial cells when we just pump red blood cells versus red blood cells with 100 micromolar hydroxyurea, you see a significant increase in the amount of nitric oxide in the endothelium, suggesting that the HU is stimulating nitric oxide in the endothelium somehow. We know it's nitric oxide in the endothelium because when we add L-name to the endothelium, you see here in the orange bar, we can knock down the nitric oxide production. Interestingly, if we add the L-name to the red blood cells and inhibit the red cells' ability to make the nitric oxide, we also see a significant decrease then in the endothelium NO. When we put it in both the red cells and the endothelium, we don't see any nitric oxide. And again, to prove that this is an eNOS-related mechanism, eNOS needs calcium calmodulin for activation. We do this in a calcium-free buffer, and again, you see no nitric oxide produced in the endothelium. So again, some really nice work here to demonstrate that the downstream effect of this hydroxyurea may actually be an increase in endothelium-derived NO. And again, our group isn't the first to suggest that nitric oxide has been important in sickle cell. In fact, there's some very nice NMR work and EPR work over the past few years showing that NO, again, is a major determinant in sickle cell. Okay, and does that still say I have 2 minutes left? Or?
Yeah, unless you don't want questions.
Huh?
If you don't want any questions.
All right, let me just show you one thing here real quick then. Another project that we're going on, that we're currently undertaking too, is we're looking at stored blood in our lab. And again, one of the reasons that we're interested in stored blood, so again, I don't know how many of you ever donated blood, but basically, they stick you and your whole blood goes into this bag, and this bag contains about 70 milliliters of this solution called CPD, which is citrate, phosphate, and dextrose. The citrate is an anticoagulant, the phosphate's for buffering, and the D-glucose is the cell's food for the 35-day storage period in Europe, 42 days in the US. Interestingly, after they collect the whole blood, it's simply centrifuged. They remove the plasma, and the red cells go into another solution called AS1, or additive solution 1, which is very similar to the CPD with just a couple extra added components. I want to show you something here that, again, anybody who understands a little bit about diabetes and glucose levels in the bloodstream may be shocked to hear this. I helped review grants at the NIH a couple years ago, and this is where I discovered this. The amount of glucose right now in me, hopefully, is about 5 to 6 millimolars, my blood glucose level. The concentrations of the glucose in that collection solution is nearly 130 millimolar, and in the storage solution is about 110 millimolar. And again, anybody who studies protein glycation or glycosylation, et cetera, is going to tell you that that is going to gunk up a cell's machinery and ruin it. Okay? And so one of the things that we've done in our lab is we've actually taken a look at preparing this CPD and the additive solutions in exactly the same manner that they use now, but just using 5.5 millimolar glucose instead of the 100 and some that's currently used. And we measured the amount of ATP coming from the red blood cells. And so here's the ATP release after day 1 of storage, day 4, 8, for the current CPD-AS system. By the way, these values right here, even on day 1, is the exact ATP release values that you see from people with type 1 and type 2 diabetes. Very low. Okay. Here's the ATP release from the same cells but stored in the normal glycemic values. And again, these are very typical of a healthy patient, the ATP release values. Again, I didn't really like the decrease here, so we've actually fixed this data now where we periodically feed the cells and keep them at 5.5 millimolar. We can now keep the ATP release values from these stored cells the same all the way out to day 36. So essentially we've got ATP release values from our stored blood that's as good on day 36 as it is the day that it's drawn from the human body. But again, since this session's about nitric oxide, I wanted to show you one other thing. We use these stored cells to look at the amount of nitric oxide that was being produced in an endothelium where stored blood was flowed underneath these endothelial cells. This is the nitric oxide produced from the system that's currently used with the high glucose. This is the nitric oxide that was stimulated in the endothelium by red cells that were stored in our lower glucose systems. You can see here, although normalized, it's about a 3-to-1 ratio of NO for the two systems. If you go back and look at those ATP release values on day 4, which is where that NO was just measured, again, about 220, 230 to about 80, almost the same ratio. So we really think that we have something going here with the stored blood and the ATP and nitric oxide story. So again, I'm going to stop talking now. Again, some conclusions. I think that the insufficient nitric oxide bioavailability can be investigated very well in a controlled manner with these microfluidic devices. And again, we've already looked at sickle cell transfusion medicine. And we're trying to do CF, but the mice, they do not have much blood in them. And so that's a problem. Again, Mark mentioned that as well in his talks, talking about sample sizes. So a good analytical problem. And again, I think one of the things, though, that my group's going to have to do over the next year or two is we're going to have to improve our tools to really control what's going on or what's affecting the cells before we can really make some solid conclusions. And so I want to thank Merck for helping support some of the work that I presented today, and also the Spence Group researchers. Here's a recent picture. This is Jada here, who's making the oxygen control systems. I mentioned a little bit about the MS project. That's Suzanne. She's working on that. Who else we got here? Steve's been key in a lot of our electrochemical detection schemes and also our microfluidic device. Imung is doing a lot of the stored blood, and Sarah worked on a lot of the hydroxyurea work that I showed you today. So again, thanks for listening, and I'll take any questions.
Questions for Dana? Yeah.
Dana, for equilibrating oxygen with the flow stream, Teflon membranes are practically transparent to oxygen, so why go with titrating with sulfate? Just have a controlled gas stream, a few microns of membrane like you have in a Clark oxygen electrode, and just equilibrate that.
Right. Yeah, we've thought about different schemes. Having sort of even an oxygenated or deoxygenated chamber, doing it that way. The other thing too that we're starting to discover is again PDMS, which again I love PDMS. It's helped us do a lot of things that we'd need a much more sophisticated lab to do. But now that we're working on some of these polystyrene-based devices, we're seeing that measuring the the oxygen levels. Yeah, it's got oxygen in it, and when we start depleting it, it repletes it into the stream. And also, we've tested a couple drugs with PDMS, and these drugs the Plavix, which prevents the platelet aggregation, that just partitions right into the PDMS. So that's one of the other things that we're looking at is membranes and materials to make sure that we really have a grasp on what's happening. Yeah.
Oh yeah, Mark one.
Real quick, why is the glucose level so high in blood storage?
Okay. So anyway, I asked when I was at this review panel, because again, I don't know anything about transfusion medicine, so I asked the guy next to me who was the director of the state of Oklahoma's Health Science Center and their blood bank director, and this guy was in his 70s, and I asked him, I said, Ron, why do they put so much sugar, in this stored blood? And his answer was, he goes, why is that a lot? And I said, yeah, that's a lot. And he's like, this is his answer, Mark. He goes, I guess it's their food. That was the answer. And so we're finding that when we put these red cells into that 100 and some millimolar solution, we'll put the red cells in there, immediately centrifuge, and draw off to measure the supernatant. Because you think there'd be, 110, 108 millimolar glucose in the supernatant? There's nothing that happened metabolically that fast. We only find about 40. The glucose molecules, I think, are just going right on the cell membrane. And after about 2 weeks, we do start to see enhanced glycation end products. So we're working on that right now. Yep. All set? Yep. Okay. Our last speaker here will be the main worker of the session.